Today we will talk about an application in tissue engineering. So, this is a vascular tissue engineering. So, the idea for this is to try to bring together all the basic fundamentals which we have looked at. Okay. So, let us discuss vascular tissue engineering, which is one of the applications. So, I am not going to discuss any particular paper. However, what I am going to do here is give an overview of how an application should be approached when you are talking about tissue engineering. You will have to see and understand some of the biology, and then try to come up with a design for how you would tackle the problem. Okay. So, we will talk about vascular tissue engineering because that is one of the popular areas which people are researching. And first, when we talk about vascular tissue engineering, we need to understand the anatomy and physiology, which is very basic to start with, right. So, there are different parts which are involved in the circulatory system. So, the functional parts which are involved are the arteries, arterioles, capillaries, venules and veins. Okay. So, I have put them in this order because an oxygenated blood first enters into the arteries, from where it branches out to enter into the arterioles, and then it goes into the capillaries where oxygen transfer would happen, and the deoxygenated blood is collected by the venules, which then enters into the veins, and this vein takes it back to the lungs and the heart and so on. Okay. So, this is the overall structure. So, the arteries transport blood at a very high pressure. Arterioles act as control valves through which the blood is transported to the capillaries, because with that high pressure, if it cannot enter into capillaries which actually have very thin walls. So, capillaries job is to ensure there is exchange of fluid, nutrients, electrolytes, hormones and any other substance that might have to be exchanged. And venules collect the blood from these capillaries, and veins are the conduits which transport the blood from these tissues back to the heart, and so that it can be pumped back again. So, this is your heart. So, this is the detailed diagram which shows all the heart valves and all the arteries and veins which are coming out of the heart. So, we will not bother too much about the heart anatomy and physiology here, because we are only looking at the vascular tissue engineering, not really about cardiac tissue engineering. So, we are not trying to engineer the heart, but it is important to understand which blood vessels are actually coming out of this heart and which blood vessels are entering are supplying blood to the heart. So, that based on that you might have to engineer them appropriately, because they will experience different kinds of pressures and flow patterns. Okay. So, if you were to look at the pressure which these blood vessels experience, it is not uniform. It is not just because that you have systolic and diastolic pressures, the pressure is also varying based on which blood vessel you are looking at. So, if you are looking at an aorta, the pressure is quite high, you are looking at to have close to 120, 80 to 120 mmHg, which is experienced by many arteries. But as it comes into arterioles, you can see that the pressure is dropping, and in capillaries the pressure is even lesser. And your venules and large veins and the vena cava all have much lesser pressures. Okay. So, basically your this is because the heart is pumping the blood into the aorta, and because of this force there is going to be a lot of pressure on the blood which is flowing into the arteries, and as it goes into the arterioles which are smaller, they act as control valves reducing the pressure, and beyond that there is no thrust which is pushing the bloods, right. So, it is not being pumped in any way, it is just flowing along. So, because of this the pressure for these blood vessels is lower. So, if we are talking about vascular tissue engineering, we need to understand what these blood vessels are, what they are made of. A vascular system which is composed of arteries, capillaries and veins, you mainly look at arteries when you are talking about like engineering these vascular tissues. This arterial system conducts oxygenated blood from the heart to the peripheral tissue. It contains 3 regions. So, the artery basically has the innermost region, which is the tunica intima, and you have the middle region, which is the tunica media, and the outer region, which is the tunica adventitia. So, the tunica intima is what is exposed to the lumen, which means that is what is exposed to the flowing blood. So, whereas your other tissues are not exposed to blood unless there is going to be a damage to the tunica intima. Okay. So, aorta is the largest artery, so which is the one which originates from your heart, and this aorta then starts bifurcating, and it bifurcates repeatedly to form multiple arteries, which then form arterioles and so on. So, muscular arteries also contain the same 3 layers, but they have only one elastic lamina. Smaller muscular arteries are called as the arterioles. Okay.
The muscular arteries, they do they have intima, media?
Yeah. So, they have all the 3 layers. So, basically these 3 layers are present in everything. So, the intima, media and adventitia are present in all arteries. However, in aorta and these larger arteries, what happens is this media is much thicker. So, it has multiple layers of smooth muscle cells, and it is very thick because that provides the ability for it to withstand higher pressures. As it becomes you move into muscular arteries and arterioles, this layer becomes thinner. So, you just have one layer instead of multiple layers of smooth muscle cells in your tunica media. So, capillaries are the smallest blood vessels which contain a single layer of endothelial cells and a subendothelial basal lamina. It does not have anything else. Why do you think this has to be thin?
Easy exchange of.
So, you want the nutrients and other things to be transported. So, you want to make sure that there is no diffusion limitation. For this reason, it has to be thin. So, a venous system consists of venules as well as small, medium and large veins. So, the veins are basically again composed of tunica intima, tunica media and tunica adventitia. However, the compositions of these regions are different. Intima and adventitia are similar to arterial structures. However, the media contains loosely organized elastic fibers instead of tightly packed as you would see in an artery. So, veins also contain a single layer of smooth muscle cells in contrast to the multiple layers which you see in arteries. So, this is all dependent on what kind of pressures these are facing, right. So, arteries, large arteries especially face very high pressures. So, the structure is designed to be appropriate for that. However, the smaller veins do not face that kind of pressure, or even smaller arterioles do not face that kind of pressure. So, obviously, their structure and organization is going to be slightly different to fit the needs of that tissue. So, when you are talking about cells, there are 3 major cells which people look at. So, you have the tunica intima, which is the innermost layer, which is nothing but an endothelial cell lining. So, this endothelial lining basically prevents blood from getting activated, platelets from getting activated. It maintains blood homeostasis. Okay. So, only when there is a rupture in this endothelial cell lining, you will have blood coagulation and wound healing cascades starting. Okay. So, smooth muscle cells are the cells which are aligned in the tunica media. So, here these are very tightly packed, and these cells constitute the major component in that region. And finally, you have the fibroblasts which are present in your tunica adventitia. So, this tunica adventitia has fibroblasts, but it is not as dense as you would see in the it is not as dense as the smooth muscle cells in your media. Okay. So, these are the 3 cells which people have to work with, and you might have to arrange them in that particular structure to get the tissue to emulate what your engineered tissue to emulate what the natural tissue is. So, endothelial cells are nothing but monolayer epithelial cells that line the blood contacting surfaces of the blood vessels. They express specific surface receptors which include von Willebrand factor, which is VWF, and VEGF receptor 1 and receptor 2, and also factor 8. So, these are all involved in different aspects of either angiogenesis or blood homeostasis. So, they allow selective transport of plasma substances and molecules, they regulate coagulation, they regulate leukocyte transmigration, contractility, vascular cell proliferation and migration. So, that is the role of the endothelial cell lining in your blood vessels. So, smooth muscle cells are basically fiber like muscle cells which are found in the tunica media of arteries and veins. They are aligned in a circumferential direction of the blood vessels, and arteries actually contain multiple layers as I was saying, whereas veins contain a single layer, and this multiple layers in the arteries are organized between the elastic laminae. So, that provides the elasticity for the material, for the tissue. Veins contain a single layer which is found right underneath the intimal layer of the venous wall. So, just below the endothelial cell lining you have another layer of smooth muscle cells. So, this is actually just defined by the location in the tissue. So, it is because they are present in the media, they are the smooth muscle cells. All the cells which are localized in this region are classified as smooth muscle cells. So, some of the markers which are used to identify smooth muscle cells are alpha actin, smooth myosin 1 and calponin. So, they allow for vessel contraction, relaxation and regulate vascular cell proliferation and migration. They are also involved in ECM secretion and production. Fibroblasts are the last set of cells which are found in the tunica adventitia, and these are found as randomly organized cells, and the cell density is significantly lower as I was saying. They act to produce the ECM and regulate vascular cell proliferation. They can synthesize type 3 collagen and other proteoglycans. They also regulate the function of vascular cells through growth factor production. So, some of the growth factors which are produced are fibroblast growth factors and epidermal growth factors. Okay. So, so this ECM of the blood vessel is just like many other ECMs. You have collagen and other proteoglycans, and but they also have a lot of elastic fibers to provide the elasticity, and this ECM provides the structural support, mechanical strength and the elasticity for the tissue. It plays critical role in development, morphogenesis and pathogenic remodeling. So, basal lamina is the thin membrane which is found underneath the endothelium, which serves as the supportive substrate for the endothelial cells. So, it cannot just be cells, right. What we looked at 3 layers. So, the tunica intima we said had endothelial cells, but these endothelial cells have to adhere to something. So, that is your basal lamina, and basal lamina primarily consists of type 4 collagen, fibronectin and laminin. And when there is an injury or endothelial attachment, the basal lamina initiates platelet and leukocyte adhesion, which triggers the blood coagulation cascade. Vascular collagen matrix is composed of primarily type 3 collagen. It also allows for the smooth muscle cells and fibroblasts to attach, proliferate and create the required patterns. So, the elastic fibers and the laminae which is present provide the elasticity. Proteoglycans provide the structural assembly and organization of the cells. So, each of these have their own importance and in the way the tissue is developed. Sorry. Okay. So, there is regulation of blood flow in when it comes to these tissues. So, you have to have blood vessel, you have to have blood flowing in one direction. So, there are local signaling molecules which are generated during the metabolic processes, which stimulate relaxation or constriction of the smooth muscle cells, and this also augments this provides an resistance to the blood flow, thereby regulating the blood volumetric flow rate. So, some of these metabolites include carbon dioxide, hydrogen ions, lactic acid, AMP and ADP. So, when you exercise, you can actually increase these metabolite production, thereby you can induce dilation and increase blood flow to the heart and other skeletal muscle system. Arteries are also innervated with sympathetic nerves. Their main function is to secrete norepinephrine. So, norepinephrine, which binds to these binds to receptors, can actually induce smooth muscle cell contraction. So, they can when it binds to some other receptor, the beta receptors, it can actually induce smooth muscle cell relaxation. So, this helps in regulating the blood flow. It helps make sure that there is a pulsatile effect which causes the blood to flow. So, exercise can again cause norepinephrine secretion, therefore create redistribution of blood flow, because you might have more of more pulsatile movement of your blood vessels. So, the blood flow itself is driven by the arterial blood pressure, which is produced by the beating of the heart. However, this pressure is also highly regulated by other receptors like baroreceptors and chemoreceptors, central nervous system, hormones and the vasopressin system and so on. So, baroreceptors are located in the carotid sinus near the carotid bifurcation, and they sense mechanical stretching due to arterial blood pressure, and increased blood pressure can stimulate baroreceptors, inducing action potentials that are transmitted to the central cardiovascular control, which will then reduce the activity of the sympathetic nerve system, so that there is a dilation and reduced blood pressure. So, chemoreceptors are similar in location and function, but they sense decrease in oxygen concentration and increase in carbon dioxide concentration instead of looking at mechanical stretching. So, the signals which they read are different between chemoreceptors versus baroreceptors. So, why do we need to look at vascular tissue engineering? So, atherosclerosis is actually a very common disease condition. So, this is a progressive development of atheroma, which consists of ECM, cholesterol, calcium, smooth muscle cells, macrophages, and it is covered by endothelial cells. So, this is a plaque which gets deposited, and it is found in the lumen of the large and medium sized arteries. So, this will basically create a block which prevents blood flow. So, which can cause serious complications, right. So, the growing atheroma can partially or completely block this lumen, depending on how big it is and at what stage you are looking at. This can happen in different parts of your body, which can cause different kinds of complications. If you are having it in your brain, you might end up with a stroke. If you have it in your heart, you end up with a myocardial infarction. If it is in the kidney, you might have renal infarction, and if you have lower limbs, it can lead to ischemia. All of these can actually cause serious tissue damage and even loss of life.
Doesn't this happen in small diameter vessels?
It does. So, but see, in small diameter vessels, if one vessel is blocked, it is usually not that big a problem, right, because you would have multiple capillaries, and even if one of them is blocked, it may not cause very serious ramifications. However, if your large blood vessels are blocked, large arteries are blocked, it will be a serious problem, because supply to many regions will get affected. Okay. So, atherosclerosis has been identified to have a lot of correlation with endothelial dysfunction, and when there is an endothelial damage, what happens is endothelial permeability changes, and cellular adhesion also changes. So, you have more fibroblasts and smooth muscle cells adhering there, causing this kind of a plaque deposition. So, that is why a lot of interest has been growing with respect to understanding endothelial dysfunction and atherosclerosis, and looking at how diabetes affects endothelial dysfunction, which can in turn cause heart disease. So, all these correlations are being studied to try and understand what is actually the cause. So, that is the basic biology side of it. So, our goal is to look at treating this, right. So, conventionally the treatments are using anti-hyperlipidemia agents which control the lipid levels. You just take tablets or anti-proliferative agents which will prevent smooth muscle cell proliferation. You can take vasodilators which will improve blood flow to the heart and reduce ischemia. You can also take beta adrenergic antagonists which will reduce the heart's work by blocking these receptors. You can end up having angioplasty where you mechanically open the arteries using balloon inflation, but restenosis is usually a concern. So, people nowadays do not do just balloon angioplasty, they go and place stents after doing a balloon angioplasty. So, stents basically mechanically opens the arteries with a metal frame and keeps it open. As long as the stent does not collapse, it will stay open. Again here restenosis is a concern. You have arterial reconstruction which is possible, where vascular grafts from autologous sources can actually be reconstructed. You can also use polymers and other materials. So, people have even tried using biodegradable polymers as for a temporary fix while the actual plaque is being treated. So, there have been many strategies. So, with angioplasty people had initially also looked at taking a fan blade kind of a mechanism and actually cleaning out this plaque. But what people realized was when they do that there is actually aggravation with plaque deposition. So, they have stopped doing that now, they primarily do only stent placements. So, these are some of the conventional treatments. There are also molecular therapies which are done which attempt to treat atherosclerosis by preventing cell adhesion, thrombogenesis or vascular cell proliferation, migration or intimal hyperplasia. Okay. So, there are different factors and molecules which have been tried. So, people try to use mitogenic factors or cell cycle inhibitors and nitric oxide promoters which have actually been used to try to prevent this formation of atherosclerosis or even reduce this problem. Okay. So, our goal for today is to propose a treatment model that will use a tissue engineering approach. So, basically we need to engineer a vascular tissue which would be useful in treating this condition. So, if that is the goal, we need to identify what would be the cells that are required, what would be the materials that are required, what would be the signals, like, so I am just putting it as a bioreactor, but what would be the signals you would want to provide to this tissue which you are creating to make sure that it is conditioned for the application you are looking at. So, this is basically what I had. So, I will just quickly go through because we are running out of time. So, epithelial cells, smooth muscle cells, endothelial cells, scaffolds obviously biocompatible, flexible and should be able to withstand contractions, and at the same time it should not collapse because it needs to be stable enough to collapse. So, if you are going to use something natural, then using a template of existing vasculature which would be your decellularized matrix, or if you are going to use synthetic, PET and Teflon would be the alternatives which are commercially looked at. So, must be rigid and flexible enough for mechanical strength to train the cells, because the cells which you are seeding may not have experienced the same kind of mechanical pressure. So, you need to condition them. So, the reactor should actually have a pulsatile flow system which can have a flow rate of about 5 liters of blood per minute and should be able to withstand pressures in the range of 1 to 5 kilopascal. So, it should also have media exchange for long term growth because you are going to have multiple layers. The other aspect is people usually work with drug eluting stents. So, you could also look at loading drug molecules to these scaffolds, right. So, which could help in anti-inflammatory properties and ensure there is better host integration. So, that is also an aspect which people can look at, but there will be challenges with respect to this being weaker than the natural muscle. There can be immune response and there can be further damage to the tissue while it is being placed. So, these are some of the challenges which have to be accounted for.